Hello, and thank you for joining us today. Welcome to our webinar entitled Measuring Helicase Activity with Transcreener and Heliscreener, Biochemical Assays for Screening and Inhibitor Profiling. I'm Justin Brink here at Bellbrook Labs. Please note that if you have any questions during the webinar, you can type them into the chat box. We'll do our best to answer them during the live discussion at the end of the webinar. Now I'd like to introduce to you our presenter, Dr. Bob Lowry, founder and CSO at Bellbrook Labs. Bob has a PhD in biochemistry from the University of Wisconsin. At Bellbrook, Dr. Lowry has led the development and commercialization of the Transcreener HTS platform for group transferase enzymes, as well as the Aptifluor SAH methyltransferase assay, the first aptamer-based HTS assay of its kind. Dr. Lowry's current studies are focused on assay development for enzymes involved in the innate immune response and DNA damage repair, including the Heliscreener assay that we'll be discussing today. Thank you, and with that, here's Bob.
Thanks, Justin, and thank you all for joining. And we'll get going here today. We're going to be talking about using the Helicase assays developed here at Bellbrook Labs based on the Transcreener HTS platform, and new assays for detecting their unwinding activity called a Heliscreener. So to give you a summary of what we're going to be talking about today, we'll be going through a brief overview of dead box helicase function and biology and the reasons why they're being therapeutically targeted, which I'm sure some of you know a lot more about than I do. And then we'll get into how we measure the ATPase activity using the Transcreener ADP2 assay, and then spend more time looking at the recent assays that we developed for detecting RNA unwinding called Heliscreener. And look at some of the details there, when the applications where you want to use endpoint mode versus kinetic mode with that Heliscreener assay. And then we'll also look at situations that might be where you might want to make a choice as far as which assay you're going to use, ATPase or unwinding. And some of the messages that I'll be imparting there, that compounds can have significantly different potencies in the two assays, and give what I think is a molecular explanation for that based on the mechanism of these enzymes. And really, the bottom line is that we think that both assays should be used in developing dead box helicase inhibitors. And some of the people who are working most intensively in drug discovery in this area seem to agree with that. So dead box helicases, they bind and remodel nucleic acids in an ATP-dependent manner. So they are helicases in the sense that they cause the unwinding of RNA, but they're not the prototypical helicases that you think of, the processive DNA helicases, where they bind at one end and then process down the nucleic acid strands. So it's more of a one and done type of mechanism where they bind and they hydrolyze ATP, and it's not necessarily stoichiometric. They can hydrolyze a lot of ATP to displace a single strand, and then that by distorting the RNA, they actually cause it to displace. There's also some studies out there showing that some of them can use more of a molecular winch mechanism where they kind of draw one strand through, and so in that sense are somewhat processive. But in any case, they function quite differently than the DNA helicases, they're the largest class of enzymes involved in RNA metabolism. There's more than 70 members, so there are a lot of these. It's not really clear that they've all been discovered yet. And included in this classification as dead box helicases are two subclasses, and the most of them have the most common DEAD motif at their catalytic center, but there's another subclass called DHX helicases, which have a slightly different motif at their catalytic center. And there are some differences in the way that they function, but we won't get into that in too much detail. I'll be referring to them all as dead box helicases or even just as DDXs for simplicity. They don't have sequence specificity, so they bind non-specifically to RNA, but they do have some requirements for the termini. And most notably, the DHX subclass of helicases seem to prefer a 3' overhang. I'll just mention that some of them are also functional with DNA. So it's a little bit like we tend to do, we want to classify everything, but for the most part, these are RNA helicases. And, most significantly, really, for the perspective of this seminar, is that their overlapping roles in RNA metabolism and as immune sensors for viral, bacterial, and self-RNA make them pretty attractive as drug targets. So just to give you, the big picture on their involvement in RNA metabolism, they are involved in virtually all aspects of RNA metabolism. To give you a couple examples, so DDX2A was actually, before it was known to be an RNA helicase, was called EIF4A, so eukaryotic initiation factor 4A. And its role is to scan down the RNA in front of the initiation complex to clear any secondary structures so that the initiation complex can continue to move down the mRNA and find the initiation site. And it's kind of an interesting one because the first DDX helicase inhibitors that I'm aware of were actually natural products, natural insecticides isolated from plants that were developed as cytotoxics. And they lock this DDX2A into a stationary complex on the RNA, so they prevent its functioning in translation initiation. Another example in RNA metabolism is DDX6. It's a very different role where it catalyzes the insertion of RNA into condensates in the cytoplasm, so essentially sequestering and taking them out of the action. So in that case, it's actually regulating gene expression. And many times, especially in RNA metabolism, the RNA helicases function by affecting the binding of another protein to the RNA strand. So very diverse and prolific roles in RNA metabolism. They also function, overlapping roles here as immune sensors. And for nucleic acids, mostly RNA, but DNA as well, for many viruses and bacteria. And the big picture of that is shown on the left, where you have viral nucleic acids entering the cell and interacting with a sensor like a DDX protein, then binding to other proteins, activating transcription, getting a type 1 interferon response. That occurs with bacteria. It also occurs, or viruses. It also occurs with tumor DNA. So you can get immunity to cancer, and it also can get hypersensitized and cause problems when it's overreacting to self-nucleic acids, and then you have an autoimmune condition like lupus. So this is the big picture here, and the purpose of this is not to commit this to memory, but just to show you that these proteins, these RNA helicases, dead box helicases, are involved in deeply entrenched and involved in pretty much all aspects of the immune response to nucleic acids, which because of the overlapping roles and the yin and yang of the immune system, so you want them to respond to things like viruses, but if they become hypersensitized, then you have an autoimmune condition. Similarly, you want them to respond to tumor DNA, but they can also, respond in a negative fashion and actually act as immune checkpoints. And I'll give you an example of that, of a drug that's actually in the clinic. So deeply involved in as immune sensors, and that places them as therapeutic targets for antivirals, for anti-cancers, and for autoimmune conditions. So some examples here of where they're being pursued or validated as drug targets. And this is an example that I mentioned, the company Accent Therapeutics now, which has a DHX9 inhibitor in the clinic. I believe it's in phase 2. And it actually turns out to be an immune checkpoint. So it dampens the response, the type 1 interferon response to tumors. So they're inhibiting that to, improve the effect of immune checkpoint inhibitors. Another example here where DDX11 is essentially acting as a synthetic lethal, or is a synthetic lethal, in this case in breast cancer. And so by combining a DDX inhibitor with PARP inhibitors, you can selectively target those cancer cells. And most of the reason that they're being targeted as antivirals is because they're host proteins that are hijacked by the viruses to enable their replication or their translation or whatever. And the best example of that is with DDX3, where it's been shown to interact with HIV-1 proteins and viral transcripts to promote the translation. It also supports SARS-CoV-2 replication, and there's a paper out there showing a DDX inhibitor being used to decrease the SARS-CoV-2 load in clinical isolates. So being targeted for antivirals, mostly cases where it's being hijacked. It also, I mentioned, is involved in autoimmune conditions. So here are a couple papers here actually targeting RIG1, which is also known as DDX58. And in these cases, it's being targeted for macular degeneration, and then also for inflammation in sepsis-associated acute kidney injury. So, important autoimmune, oncology, and the antiviral indications for these targets, which is why we became interested in them. And I assume why you are as well. So the assay methods that I'll be talking about today are, first of all, the Transcreener ADP2 assay, which we use to detect the ATPase activity of these enzymes. And I'll go into detail about how it works later, but suffice it to say, both these assays, they detect the core catalytic functions of the RNA helicases. So the unwinding or the dissociation of one strand from the other of RNA requires, the energy provided by ATP hydrolysis. But the thing to keep in mind as I go through the talk, and I'll bring this up again, is that these two functions are not tightly coupled. That is, it's not always that efficient. In other words, it sometimes requires, many ATP, as many as 100, to be hydrolyzed to displace a single strand of RNA. And that's dependent, as you would intuitively sense, on the length of the RNA being displaced. So as you go out to longer RNAs, it requires a lot more RNA to displace that. And again, it's not because it's a processive mechanism. This is a conformational type of mechanism where it actually has to bend the RNA to cause its dissociation. But what happens is it gets into futile cycling. It makes a lot of tries and it's not always successful. So these are the two assays that we'll be talking about. They're both mix and read assays. They're both fluorescent readouts and designed, of course, like all our assays, for high throughput screening. So this is a brief schematic of the Transcreener ADP assay. It's a very straightforward, competitive immunoassay. So ADP binds to a highly selective antibody, which does not recognize ATP. It displaces a fluorescent tracer, which changes its fluorescent properties. And we've developed assays for different readouts, fluorescence polarization, TR FRET, and fluorescence intensity. But they all function basically the same way. It's the only assay capable of directly detecting ADP produced, without the use of additional coupling enzymes or something like that. It's the direct detection of ADP, and it's also the most sensitive assay for ADP detection. And it's been extensively used for more than 15 years for screening kinases and other ATPs, so it's highly validated for high-throughput screening. So this is the type of optimization we do for the Transcreener ADP assay. This is after, optimizing the RNA concentration and ATP and doing a fair amount of research on the best RNA substrates to use. So the conditions that we decided on, which are functional for all of these DDXs shown in the table on the right, we're using saturating RNA concentration, which is under the assumption that people don't want to find molecules that are competitive with RNA, because a lot of those turns out to be intercalators. But if you want to do that, you can use lower ATP concentration. We also use subsaturating ATP, which is around 100 micromolar for most of these enzymes. Again, because the assumption being that you do want to detect ATP competitive inhibitors. But again, those parameters are up to the user to change if they want to. We tested probably about a dozen different RNAs of various structures with different types of ends and all that kind of thing for these assays. And we actually found that just bulk yeast RNA did the best, catalytic turnovers with all of the enzymes, with the exception of RIG1 and MD5. So, so that's what we're using for all of the classic dead box helicases. Now, again, if you want to use a specific RNA substrate, you're free to do that. You can measure ATPase with that just fine. But the way we've set these assays up is with the total yeast RNA. So you can see here we've done an enzyme titration here with 3 different RNA helicases. You can see that's completely dependent on the presence of the RNA. So you don't see ATPase or very, very little in the absence of RNA. When you convert that polarization signal to ADP formation, you see you get a nice linear response to the enzyme concentration, which is what you want to see. The table on the right shows the typical enzyme concentrations that we recommend to generate this type of initial velocity response, which is up here, about the EC80 on these titration curves. So you can see that for most of them, it's low nanomolar. For some of it's nanomolar. DDX41 is one of the slower enzymes. But all of these are low enough that you can measure easily the potency, accurately measure the potency of nanomolar inhibitors. And also that, you're not going to be running through a ton of enzyme in a screen that requires a lot of wells. So we think of those things when we optimize these assays. Let's just mention briefly that RIG1 and MDA5 have to use specific substrates. They don't function well with the yeast RNA. So this shows some pilot screens that we ran using the ADP2 assay for ATPase activity. This is a DDX5 pilot screen, and you can see that most of the compounds cluster very tightly around the uninhibited controls, which is what you want to see. The result of this screen was 15 hits. This is a 1280 compound library of bioactives that we use for these pilot screens. 15 hits. We always triage interfering compounds. We run a mock screen. I'm setting up an assay that looks like a completed enzyme reaction, but with no enzyme present. And from that, we found 3 interfering compounds. And you can see the screens that we ran with all of these were very good, robust assay windows, Z primes, most of them around 0.9, with the exception of DDX41, which is still above 0.7. So quite robust for high throughput screening. This is an example of using the assay in dose response mode, using a couple of hits from the DDX5 screen, and suramin as a positive control. Suramin is a very promiscuous compound that we use basically for all nucleic acid binding proteins. We find it inhibits almost all of them. And you can see we get a nice dose response curve, from which you can calculate the IC50s and rank profile your inhibitors. So that's the ATPase assay. Again, we've screened a number of these, run pilot screens for a number of these helicases with that, and it functions very well, both for the screening and the dose response. So for the unwinding assay, this is a fluorescence dequenching assay, and this type of assay has been used for helicases, more for DNA helicases for many years. But our goal is to develop an assay with the performance required for HTS, and so a nice, good assay window and very little noise. And that was somewhat generic for a number of different DDXs. So we ended up, we tested a lot of different RNA structures, and different fluoroquencher pairs, different labeling strategy, versus a panel of DDXs, 7 different DDXs. And we found that this configuration was generic. It worked well for 4 of those DDXs. So it's a double-stranded RNA, really a triplex, with a 3' overhang, and then labeled here at the joint point with an Iowa black quencher and a Sci5 fluor. And so when the RNA helicase causes displacement of that strand, you get the dequenching and you get a nice fluorescence response. And then we use this capture strand here to prevent re-annealing of the fluor to the template. So it's a stable signal. So we optimized a lot of things: RNA annealing, how we store it, the order of addition, all those things to make a difference, and the buffer and additives, ATP and RNA concentrations, and defined protocols for both endpoint and continuous assays. And as you'll see, our recommendation is that you use the endpoint mode for screening, just because it's a lot more convenient. But to get accurate IC50s, that you use the continuous assay or kinetic assay, and I'll show you why that is. So the protocol, like all of Bellbrook's assay, is designed to fit into an automated HTS workflow. So it's mix and read. You had your enzyme, your ATP, you had the RNA, the fluorescent probe, and then you run your reaction, and for kinetic mode, you monitor it periodically, read the fluorescence, or for endpoint mode, you had a quench mix, which contains EDTA. These are all magnesium-dependent enzymes. And you can do that then as an endpoint single read assay. And the only real difference in the components in these assays between the different isoforms is the ATP concentration. Again, we're using subsaturating ATP with the assumption that you're going to be interested in ATP competitive inhibitors. If you're not, you can use 5 millimolar ATP, and most of those you won't find many ATP competitive inhibitors, and the reactions will actually run faster in most cases. I'll just note that these ATP KMs for the unwinding reaction are actually different in some cases than they are for the ATPase assay. So this is an example of what the data looks like. This is the 4 different RNA helicases for which we've optimized this assay. So you can see that with increasing concentrations of enzyme, you see an increase in fluorescence as that labeled fluorescent strand is displaced over time. And so for the endpoint assay, we recommend, we ran endpoint assays at 15 minutes. That's a convenient time point. And we recommend using an amount of enzyme it's in most cases, it turns out to be between these 2 curves here. So it's not up here where the curve is flat, but it's beyond the linear range, as you can see. And the reason that we did that, we're typically real picky about running things in initial velocity, but in this case, you can see at these lower concentrations, some of these they really never reach this saturation, this maximal window, and we don't know if the enzyme just poops out or what. So we recommend using a concentration enzyme to get you fairly close to this maximal signal, because that's really what you want for high-throughput screening, even though it's not in the initial velocity region. And so that's where these enzymes concentrations were drawn from. So you can see for DDX5, we're recommending 5 nanomolar. So that's right in between these two. So it's up here out of the linear range, but it's definitely not where it's flattened out. The enzyme is still functional there. And using concentrations of enzymes, in that region, you can see that you get very good separation of positive and negative controls, and your Z primes, all of these are right around 0.9. So it's a very strong, robust assay window in this endpoint mode. So this is an example of a screen, and for DDX17 run in endpoint mode. And again, you can see very good tight clustering, and the Z prime on this was right around 0.9. So, in this screen, actually, we identified a number of inhibitors as well as activators, which are interesting. This compound up here, I wouldn't pay too much attention to. That's almost certainly interference if it's up that high. Again, we can run a triaging, a counter screen for interference. And we did observe higher levels of interference in this screen than we typically do in a Transcreener assay. And this is N of 1. We haven't run this assay in screening mode for any of the other isozymes other than DDX17. So we don't know if that's a generalization or not. But in any event, they're very easily to identify. These are dose response curves of a couple inhibitors, a couple of the hits identified in that screen. And you can see we get pretty good responses. You always get some oddballs like this when you're running high-throughput screening. This is probably not a compound that we would follow up on, but we do get a dose response. So it was a positive from the screen. And actually, both of these were confirmed in the ATPase assay as well, I might mention. So when to use the endpoint versus kinetics for a high-throughput screening, I mentioned, for convenience sake, running the endpoint, I think, is the way to go. We don't recommend it for running dose response, and here's why. So these are dose response curves in the kinetic mode and the endpoint mode. And they look similar, but if you look at the x-axis, in the endpoint mode, it's significantly right shifted. So that means the IC50s are higher, 3 to 4-fold higher in endpoint mode rather than kinetic. And the reason for that is that you're measuring the activity out here beyond the linear range. And so it's not going to adhere to the Michaelis-Menten kinetic rules that you need. So all the assumptions won't hold. So the math just, it's not valid in this nonlinear range. That's why we always tell everybody to run your assays at initial velocity. Well, the assay is still in linear mode. So we recommend using the kinetic mode for accurate dose response measurements. And this slide illustrates the way that that's done. So you run the assays in kinetic mode, just read the plates periodically, do your normal inhibitor titration like you would, and then select, the linear part of that curve that's linear throughout the entire inhibitor concentration range. In that case, this was from about 1 to 5 minutes. And then you convert that to rates, determine your rates of the slopes of these lines, and then from that you plot your dose response curve. And that way you're going to get very accurate IC50s based on the initial velocity of the enzyme. So I've shown you 2 different types of assays, and both are compatible with high-throughput screening. Both can give you accurate dose response measurements. So which do you use when? And I can't answer for that, but for you, but I can give you some information that might, inform your decision. So the first thing to remember is that we're using very different substrates in the two assays. So in the unwinding assay, we're using, a short defined RNA with a 3' overhang. I think that template strand is only about 30 nucleotides long. And so the piece we're displacing is 13 or 14 nucleotides versus, just random RNA in the ATPase assay. So there's going to be a lot of long RNA in there and a lot of short and every kind of end. It's a little surprising to us that it actually works the best, supports the best catalytic activity for ATPase, but that's what we see. The other thing is that the ATP concentrations differ, but it's subsaturating in both assay formats. So in that case, it's somewhat, balanced out there. Because the KMs are different. So this shows you the kinetic parameters determined for the ATP KM using the ATPase assay and the Helicase assays. You can see in some cases they're pretty similar, but in some cases they're quite different. 250 versus 1350 micromolar for DDX5, or in this case the opposite. 400 micromolar versus 1.8 millimolar for DDX17. So, we're using ATP concentrations to come close to those KM values in that assay. So that's another difference. The differences in the enzyme concentrations aren't that big, and because, unless you're looking at sub-nanomolar inhibitors, they're not going to make a difference. So this is another interesting, a result that we found that could inform your decision. So we did screens using the ATPase for these 3 different DDXs, and then measured the dose response. Some of them were overlapped with some of them were specific to a single enzyme. But anyway, we tested them all in dose response using both the ATPase and the unwinding assay. And here's the relationship we found. So for a number of inhibitors, there was a pretty good correlation where the IC50 in the Helicase assay was about 2-fold lower than in the ATPase assay. So they're pretty similar, but interesting, just a bit lower in the Helicase assay than the ATPase. But then there was another subset where, they were much more potent in the Helicase assay than in the ATPase assay. And if you look at this compound in here, which takes that to extreme, the IC50 in the Helicase assay was sub-micromolar, 0.5 micromolar. So that's a pretty good hit. Whereas in the ATPase assay, it's 38 micromolar. How do you explain that? They're both measuring the catalytic activity of the enzyme, but you're getting very different IC50s. So one possible explanation for this, which we think is pretty likely, is that this compound is acting to uncouple the ATPase activity from the unwinding activity. So it could be an allosteric inhibitor where it's actually, preventing the translation of the energy from ATP hydrolysis to the unwinding of the RNA. So the conclusion that we draw from this, and you can draw your own, is that we know that RNA helicases have complex mechanisms. They undergo multiple RNA-dependent conformational changes during a catalytic cycle. And I'll add that there isn't a tremendous known about the human dead box helicases. A lot of the work has been done with yeast and other types of enzymes. And it's not clear how much you can generalize, but it's known that they undergo conformational changes. That's clear. There's structures out there for the human enzymes. And it's known that ATPase and unwinding is not always tightly coupled. We've shown that using our assays, side by side. We've seen as much as 100-fold higher ATPase uncoupling compared to DNA strand displacement. And other investigators have seen that as well. And so we believe that inhibitors should be characterized for effects on both the ATPase and the unwinding activities. And one of the one of our heroes, actually, this is a paper out of from Accent Therapeutics, where Robert Copeland is the directs this group, and he's written a couple of the best really books about using enzymatic assays for high-throughput screening and hit the lead. So when he speaks, we listen. And so this is the DHX9 inhibitor I told you that they have in the clinic. And it's describing, the hit and the lead hop, hit the lead hop. And they don't tell you which assay they actually use for the screen, but you can see the hit here. This is the compound 1, which could have come up with a better name, but and you can see that it's the opposite of the case with our inhibitors. It actually has better potency in the ATPase assay than the unwinding assay, but it doesn't completely inhibit the ATPase activity. And this was the hit. So, and every derivative of this that they developed all the way through to their lead molecule never showed it. They all showed about 70% inhibition of ATPase. And, and a lot of people would have thrown that molecule away. Certainly, if you were only using the ATPase assay, you might have triaged it. But this was their conclusion. And this turned out, was developed into their lead compound. Was that, you can't be dogmatic, and you have to use multiple approaches in characterizing your hits and during the hit to lead approach. And in this case, that was using both ATPase and unwinding assays. So in summary then, I've showed you that Transcreener, the ADP2 assay, and can be used for ATPase, and Heliscreener RNA unwinding assay can both be used for screening dead box helicases and profiling inhibitor potency. We suggest that screening with the unwinding assay can be done in endpoint mode for ease of use, but we highly recommend using the kinetic mode for dose response measurements. I didn't show you this data specifically, but if you just compare side-by-side screens with the ATPase unwinding assays, the overlap, a Venn diagram, the overlap is relatively modest. So you're going to get different hits using these two different screens. And I think that has to do with the mechanism of the reaction. I think it has to do with the difference in the ATP concentrations and the different RNAs used in those assays. But in general, compounds identified in an ATPase screen show a dose response in both assays. And from the one example of DDX17, where we did the Helicase screen, the converse is true as well. So I showed you also that inhibitor potency tends to be low in the ATPase assay compared with the unwinding assay, but I also showed you an example from the literature where the opposite was true. And we think that at least what we're seeing could be due to an uncoupling effect. So before I take questions, I just wanted to show you an overview here of our product portfolio for RNA and DNA helicases. So we have all of these recombinant helicases available, and they are validated, and in most cases available in complete solution kits, including all the substrates and buffers required for all these RNA helicases, many of which I spoke about today, as well as a number of DNA helicases, BLM, WRN, POLQ, SMARCA L1, and we have the Helicase, the Heliscreener unwinding assays for the RNA helicases that I talked about today. We've just launched, or we are just in the process of launching a similar assay, which is shown here, uses a somewhat different assay configuration, but it's the same idea, a dequenching assay for DNA helicases. So we're launching that assay initially for WRN, and early next year we'll have it available for BLM, and then we will of course extend that to the other DNA helicases, including POLQ and SMARCA L1. And at this point, I'd be happy to take any questions.
All right, thanks, Bob. Let's get to some of those questions. If you have any, remember to just chat them to us, and we'll see if we can get to them here. First question, you've tested a number of helicases thus far. Do you have plans on validating more as well?
Yeah, absolutely. I mentioned that we're working on BLM. That's we know it's functional. We're just scheduled to launch for early next year. And we'll also be extending that to PolQ and SMARCA-L1. We'll also be adding additional helicases to this portfolio. We already have 2 additional dead box helicases in and functional that we'll be adding to this portfolio. We'll be adding more of the SMARCA the SMARCA enzymes next year. So yeah, we'll definitely be expanding this portfolio to additional RNA and DNA helicases in the coming year.
Here's another one. What is the maximum ATP concentration that can be used in these assays?
So for the Transcreener, for the ATPase assay, the practical limitation because of the antibody usage becomes impractical. The practical limitation is 1 millimolar. So you can go quite high with the Transcreener ADP assay. For the Helicase assay, you could go as high as you want. There is no limitation there. So.
Here's another one. How early in a discovery project should we deploy unwinding assays versus biochemical assays?
Right. So if you're using one assay for your high throughput screen, for your primary screen, then I would, as soon as I would use the other assay to characterize those hits. And if they don't register, if you don't get any inhibition when you're using it as an orthogonal assay method, then I would be suspect of that hit. So in other words, you want to use them both fairly early.
Great. So here's another one. Can Bellbrook Labs run pilot screens or custom assay development for these novel kind helicases?
Yeah, absolutely. In fact, that's one of the things that got us into this target area. We ran a number of years ago for a company, developed some assays for a number of DNA helicases, including WRN. And that got us interested in the platform. And so for an example of what we did there, we produced the recombinant proteins for them. We validated them in an assay using probe inhibitors, and that at that time there weren't many known. And then they were doing computational design of inhibitors, so we were screening, taking their compounds from their virtual screen and running them in the ATPase assay for them. So we can certainly do that for really any enzyme, but certainly any of the RNA DNA helicases, even if they're not in our current portfolio.
Great. Thank you, Bob. That's all the time we have for today. If any other questions, please contact us by email at info@bellbrooklabs.com. A copy of this webinar will be sent within the next 24 hours for review. Also, please go to our website, bellbrooklabs.com, for our latest posters and information on many of these RNA and DNA helicase targets. Thank you for joining us. Bye.
Thank you.